The mapping of the human genome was a fearless plunge into groundbreaking science. Dr. Fraser says as a scientist, she had to take risks that allowed her to answer the questions she was asking in molecular biology at a time when the field was nascent. She dared to fail at a time when that was still an option. Her PhD and her early work at NIH was focused on G protein-coupled receptors that led to exploring the receptors at a fundamental molecular level. She asked, how do you discover new members of this protein family? And that question led her to an intersection of disciplines such as automated DNA sequencing, which ultimately put her in line to map the human genome. In this episode of Careers That Matter, Dr. Claire Fraser walks us through her remarkable career. Dr. Fraser, you have had a remarkable career. The work that you have done in genomics is extraordinary. It's been groundbreaking, and it has opened our eyes to an understanding of ourselves and the world in which we live in ways that were beyond imagination a couple of decades ago. Tell me about your career. How did you come to be Dr. Fraser?
Well, thank you for those kind remarks about my career, and I would absolutely have to agree. I have ended up doing things that I never aspired to. I grew up in a small town in Massachusetts. I credit my love of science to a freshman and high school biology teacher who was enthusiastic about the subject and wasn't afraid of it. And that sort of launched me down the path of wanting to go into science. Both of my parents were in public education, and I thought up until that point, that's probably what I would do. Again, growing up in a small town, I didn't know, and this was back in the 1960s, I didn't know anybody who had a career in research. So in my mind, the only way to marry a love of science with a career was to go into medicine. And I applied to college and was thinking about that all the way through. I actually started applying to medical schools and had gotten accepted. But in my senior year in college, I did a research project in a lab, not a laboratory course, but a real research project. I was given my own project. I was working on bacteria in Lake George, New York, and the influence of fertilizer runoff. It's kind of interesting because I sort of ended up back doing similar microbiology studies later on. But at that point, I very late in my senior year, I said, I don't think I want to go to medical school. I want to go to graduate school and pursue a career in research. They're not mutually exclusive. I didn't have the best advice at that point in time, but I made the decision. And it was very late, and I wasn't thinking about taking a year off, so I scrambled and said, well, how am I going to pick a graduate school? So there was Peterson's catalog at the time. I was dating a guy who had graduated a year ahead of me. He was working in Toronto. And so I opened a map. I opened my Peterson's catalog and said, Buffalo, New York is as close as I can be in the U.S. to Toronto, but still be in the U.S. Here's the pharmacology department. It looks like some interesting research going on. That was my choice.
So you didn't sit there going analytically, this is going to be the best for my career. No, I like that guy.
And it took me years before I was even willing to admit that that's how I selected my graduate school, because I wouldn't advise that for anybody, but it all worked out. I then had the opportunity, shortly after I finished my PhD, to move to the intramural program at NIH, and I spent almost 10 years there, and that's a phenomenal environment. Mm-hmm. There are no excuses for not doing the highest quality research. And that experience, I think, really taught me to be fearless in what I would take on. And it was during that time that automated DNA sequencing was starting to appear. And
let me stop you for just 1 second. You say be fearless. Mm-hmm. What in the realm of scientific research does that mean?
To me, when I say be fearless, what that means is not be afraid of moving in a new direction, not be afraid of trying a technique that has only just been discovered. For my PhD thesis, and I started my PhD work in 1977, it was a year after the first paper on monoclonal antibodies was published. So I decided, wow, this sounds really cool. I'm going to make monoclonal antibodies against these receptor proteins that I was interested in, to see if I could use that information to distinguish them at a molecular level.
So was it a curiosity in this that drove you forward? Because you weren't sitting there saying, I'm plotting out how my career is going to unfold.
No, exactly. It was a curiosity. There was a question, and it was just, what tools do I have at my disposal to try and answer these questions? And so it was when I moved to NIH, then the field of molecular biology was just getting started. Had I been trying to get external research funding, I never would have been able to move into molecular biology, because chances are I would have been told, you don't have the experience. I was with a group of people in the lab. We self-taught ourselves molecular biology. There was this manual, the Maniotis manual, and we self-taught ourselves. And that's what I mean by being fearless. What's the worst that could happen? It's not going to work. Okay, well, then you go do something else.
And isn't that a big component in scientific work?
It is, except when, I think that things have changed, again, certainly within the U.S., with research funding. We don't have the luxury, and you might also say the necessity, of failing and expecting that we will continue to get good research support. The NIH is reluctant to fund research that's going to fail. It's hard to publish papers on something that has failed. And
despite the fact that there's tremendous learning that comes from that failure.
Despite that fact. Yeah. So I think, unfortunately, too much of the science that gets done is safe science. And therefore, it's not necessarily breakthrough potential. It's really, it's very disheartening, but when I think back to my time as a PhD student, when I didn't know any better, and then as an early career investigator in the intramural program at NIH, where there were no excuses. You didn't have to write research grants. You had funding. You just had to do good work and publish and go in new directions. That taught me to be fearless.
Yeah.
Had I not had those experiences, I might never have been able to do what I had done, because I can think at many pivotal points in my career, there was an element of fearlessness that was required. It was really going in a new frontier, going in a new direction.
So you complete your PhD in 1977, 78?
No, I started in 77. I completed it in 81.
And that moves you into what aspect of your career? Because we know that you then move on to the Human Genome Project, but what were the elements that led up to that?
To the Human Genome Project?
Yeah. Like in your own career development,
of course.
It positioned you to be ready to do that.
My PhD work and my early work at NIH was focused on G protein coupled receptors. I was focusing on beta adrenergic receptors at the time, a subgroup of those. Well, it turns out that this is one of the largest protein families in mammalian organisms. They're cell surface proteins. They're involved in cell-to-cell communication. They're found on every cell in the body, and they're critically important. And at least half, if not more than half, of all prescription drugs on the market today target G protein coupled receptors of different types. So I was interested in these, in understanding the differences from these on a fundamental molecular level. And we know so much today, but back 40 years ago, we didn't. And so one of the things that became apparent as my work on these was developing was that these were, and this was back in the 1980s, late 1980s now. It was starting to become evident that maybe there were more than just a handful of these. Maybe this was a related protein family. So I became interested in the evolution and the complexity of this protein family. And then the question was, well, how do you go about discovering new members of this protein family? And that's where my receptor interest intersected with automated DNA sequencing, the first instrument that was introduced in '87, and I had access to at NIH. And we could then just start trying to find these receptor proteins by large-scale gene cloning and sequencing. So once you have a foot in the door with sequencing, I thought I'm going to build my career using these new tools to find as many members of the G protein coupled receptor family as I can. And then another opportunity presented itself to use these same tools to try and sequence the first bacterial genome, which we did in the mid-1990s. This was, again, a situation where everybody said, this is going to fail. You can't do this. You don't have the tools. And it was very simple. This was a large piece of DNA. We broke it into smaller pieces, sequenced each of the small pieces, and much like a jigsaw puzzle, put this all back together again to get the first complete bacterial chromosome.
And that gave you a...
And that was the beginning of all of this. We weren't convinced we could do it a second time, but we did. Mm-hmm. And then all of the funding agencies became very interested. And this was right around the time when the Human Genome Project was gearing up. And the fact that what we showed fundamentally was that you could take large pieces of DNA, break them up into small pieces, sequence those, and put them back together with bioinformatics tools, accelerated the Human Genome Project as well.
Well, way ahead of schedule.
Way ahead of schedule, yes. So none of this was planned. Absolutely none of it was planned. When I participated in the first bacterial genome sequencing project, I thought it would be a diversion and I'd go back to receptor work. But I had so much fun trying to figure out what all these genes did, what proteins they encoded, how they worked together to create a living organism. There was no turning back.
So from a going to work every day perspective, as you start to get more and more immersed into the Human Genome Project, what kind of an exciting environment was that to be a part of?
It was amazing. And now with the benefit of hindsight, my colleagues and I that were involved in this at the time, when we get together again and we've all gone our separate ways, we'll talk with such nostalgia about those days. We knew we were on the frontier. There were many times when, I don't know how else to say this, we were making up the science as we went along. That's not fabricating science. I'm talking about making up the methods. We were having to develop methods. We were having to figure out how you get from point A to point B. Nobody had done that before. And that was enormously exciting. And, it wasn't until we got a few years past the completion of some of these early bacterial projects and then the Human Genome Project, where there were anniversaries to commemorate these accomplishments, that I really took that opportunity to look back and really reflect on what an extraordinary, unique position my colleagues and I were in. It isn't often that investigators help to define a whole new field of investigation, and I feel that that's very much what we did. And I feel for myself it was actually more in the microbial genomics space than in the Human Genome Project, but the learnings from the two went hand in hand.
So from I'm going to work every day perspective, did you find yourself rushing to get to work so that you could share some insight or something that had percolated in your mind overnight while you were sleeping to go, what about this?
Yes.
And like, what an exciting environment to work in.
You're absolutely right. It's hard to describe. I've been asked many times if I plan to write a book on this, and I don't think I will. But you're absolutely right. You would take your work home with you, but in the best possible way, not as a burden, but as something that you just were so excited about you couldn't let go.
So move forward. You then you are doing work that puts you in a very unfortunate situation where you're having to identify the source of anthrax that was used in the attacks in Washington and in the United States in 2001. What's it like to find yourself in that situation?
That was another extraordinary professional experience. We happen to be working on a genome sequence of a strain of Bacillus anthracis, not the one that was used in the attack, but we were leading the field in terms of microbial genomics approaches. I was very good friends with Rita Caldwell, who is a microbiologist and was then director of the National Science Foundation. Rita reached out to me. We had some conversations about whether or not I thought that genomics could help. And I didn't know, but I said, why wouldn't we try?
Mm-hmm.
You know, we can take all the resources that we have at our disposal. Meetings were quickly convened with representatives from the FBI, and we mapped out a plan. But this gave all of us an opportunity to see science being done for the purpose of a criminal investigation. It was a very, very different situation. I used to call it, that we were doing microbial genomics in a parallel universe.
Mm-hmm.
We were on a need-to-know basis. It was a very small group of investigators that even had access to any of the samples or the data. We had to be extraordinarily careful about keeping laboratory notebooks because we were generating information that everyone would believed would ultimately be used in a criminal investigation, assuming that the perpetrator could be found.
And so, when you started the Human Genome Project, you had no idea that this would be one of the unintended benefits of having done that work. Does it give a tremendous amount of promise to the possibility of genomics beyond just the medical benefits that an individual human may experience?
Oh, absolutely. I mean, I think genomics now has percolated into every field of study. Genomics is being used in agriculture. It's being used. In fact, I had an interesting conversation with a colleague this summer on Nantucket, where the seal population is rebounding, and as a result, great white sharks are coming in, and that's sort of disturbing everyone's summer fun. And we had a really interesting conversation about whether or not we could use genomics-based approaches to identify what species of shark was attacking the seals. When the seals are attacked, they die, they wash up on the beach. So here's an instance where genomics is being used to try and figure out, if you will, the ecology of this whole system. Genomics has been used in wildlife trafficking studies. So genomics is, because DNA is a universal molecule, genomics is finding its place as an incredibly useful tool in just about every application that you can think of. And fortunately, it has become quicker and really quite affordable now. So we have this as one of the main tools we can go to in our toolkits.
So a young woman who may be watching this, and I say that on purpose because I know that still the representation of women in science is not as robust as we would like to see. A young woman's watching our interview right now and says, okay, you said that you can't be quite as fearless in science as you were when you were at the early stages of your career. What do I do to have the kind of impact from my work that you've had? What's your advice to?
My advice is to go for it, and to be opportunistic. I think these key transition points in my career that we've talked about absolutely resulted because I was always keeping my eyes and ears open for new opportunities, and I was very opportunistic. And I remember when I was in graduate school, we'd have seminar speakers come in that had done certain things for part of their career, and then they changed and were doing something else. And I never really knew how that shift happened until I actually lived it. You can set out and define a path for yourself and think, I absolutely must follow this path. I'm going to follow this path no matter what. And you can make a wonderful career of that. Or you can cast a broad net and keep an open mind and look for opportunities. And I think for me, some of the most important things have been the collaborations that I've been involved in, because with collaborations, working with colleagues, you can do so much more than any individual can do on his or her own. And I think for me, one of the best things about science is that it is really such a small international community, that we, over years of practicing science, I've gotten to know so many people that I now consider to be not only colleagues, but to be dear friends. And that makes it really special as well. And so I just think that, you have to be willing at times to step outside of your comfort zone, to push back when you think it's appropriate. But the most important advice, I think, for anybody is just to listen to your heart, or we're talking about the microbiome, listen to your gut. Because if you do that, chances are you won't go wrong.
Wonderful. Thank you very much for sharing.
You're welcome.
Sharing your life's
You're welcome.
career and work from an inspirational perspective for so many people. Thank you for the work that you